So let's take a look at problem 10.5. This is about Avery, MacLeod, and McCarty, and they are trying to figure out what is the transforming principle. You know, they have this fraction of material from a certain bacterium, and it's able to transform another bacterium, and they're trying to figure out what is it in this transforming principle, what molecule in there is actually doing the transforming. So let's review a little bit. So they're working with a certain strain of bacteria, and two different strains. One's 3S and one is called 2R, and the 3S is virulent, it kills mice, and the 2R is non-virulent, non-virulent or non-pathogenic. It doesn't kill mice. But Frederick Griffith had found that, you know, if you put 3S that's been heat killed, so normally, you know, it doesn't kill mice because it's dead, but if you inject that and the 2R bacterium into a mouse, well, the mouse will die. And then Griffith is able to isolate live 3S bacteria and 2R bacteria. And I've heard it's really easy to tell if you have 3S or 2R bacterium, because the colonies form nice smooth colonies for 3S and rough colonies for 2R. So it's very easy to tell what strain you have. Well, Griffith named the transforming principle. Well, whatever factor was actually doing the transforming of the 2R into 3S was named the transforming principle. And then so scientists for I think about 20 years later took to identify what is it in here that is doing the transformation. So we'll jump right up to Avery, MacLeod, and McCarty. So what did they find? Well, so they were working with large volumes of bacteria, so large amounts of 3S bacteria, and they isolated. Well, so they broke apart they isolated the bacteria, broke apart everything, removed most of cell walls. What else did they remove? Most of the proteins, fats and lipids. They removed all that stuff, and they filtered it. So what we have is a filtrate. We'll call it a crude filtrate. Crude filtrate. And then they take that crude filtrate and they mix it with 2R bacteria. They will find that, you know, this crude filtrate will still transform the 2R into 3S. So they get both 3. It doesn't transform all the 2R, but, you know, it transforms some of them. So they end up with both 3S and 2R. So their goal here is to figure out what is it in the crude filtrate that is transforming 2R into 3S. You know, is it protein? Is it DNA? Is it RNA? Is it a combination of the 3 or 2? And so that's what we'll take a look at next. So we can take a look at the question now. So when Avery and his colleagues had obtained what was concluded to be the transforming factor from the 3S virulent cells, they treated the fraction with proteases, RNase, and DNase, followed in each case by the assay for retention or loss of transforming ability. What were the purpose and results of these experiments? And then what conclusions were drawn? So the purpose was to, as I already mentioned, was to identify what was the molecule or the factor in the crude filtrate that does the transforming. So what did they do? So, I have some stuff left over from a previous video. We don't need that. Okay. So, they have their crude filtrate, right? So crude filtrate transforms, and they treat that with proteases. Now they removed most of the protein already, but they're not sure that they got it all, so they want to get all the protein. So they add proteases, and the proteases degrade protein. So they degrade all the protein into individual amino acids. And then they take this protein-less filtrate. They add this to the 2R bacteria, and what they find is they still get 3S and 2R. So apparently protein is not part of the transforming principle. It's not the factor causing the transformation. So the next thing they do is they add RNase, and RNase is an enzyme that degrades all the RNA. So you take a long RNA molecule, the RNase will break it down into individual nucleotides. So now we have protein-less and RNA-less filtrate. I wonder if there's a better way to say that. So filtrate with no protein and no RNA. They add that to 2R, and they have, what did they get out of that? They get 3S back again. So wow, okay, so something is still in here. Whatever transforms bacteria is still in this proteinless and RNA-less filtrate. And so we all know, probably if you were in lecture, if you looked at the lecture slides, you know the last step here, they use DNases, and DNases removes the DNA, or the enzyme DNase. I think there are a number of different DNases, but the enzyme DNase will chop up DNA into little pieces. So DNA less, protein-less, and I'm sure there's a better way to say that, but RNA-less filtrate. And when they add this to the 2R bacteria, they only get 2R back. So finally they removed whatever the transforming principle was. So whatever it is, whatever transformed the bacteria, it seems to be dependent on the presence of DNA. So this is the first really good genetic evidence that, or evidence I should say, that DNA is the genetic material. And so that's the answer to question 10.5.